Today's lecture will be on the embryology part. Mainly, we focus on the formation of gametes, how the sperm and the egg cells are produced. Because fertilization involves union of sperm and egg. So first, before we proceed to the embryology part, we try to discuss on the development of the gametes, the sperm and egg cells. So in general, human development is a continuous process that begins when an oocyte or ovum from a female is fertilized by the sperm or spermatozoa. And so in general, human development begins with a union of sperm and egg. This union of sperm and egg takes place in the fallopian tube. And once the sperm and egg are united, the sperm carries 23 chromosomes, and the egg cell carries another 23 chromosomes. The two cells unite and form the zygote, a one-celled organism. This zygote undergoes successive mitotic division and finally give rise to the human being. And this entire process, starting from the time of fertilization to birth, is called embryology. It also studies about not only the normal development, embryology also studies about birth defects and their causes. What kinds of birth defects are there during each successive development, and what are the cause of these birth defects? It could be the cause could be genetic, environmental, or mainly these two are the cause of birth defects. So that branch of embryology is known as teratology. In general, embryology, intrauterine growth, it takes about 38 weeks if counting starts from the time of fertilization. If counting starts from the time of fertilization, from the date of fertilization, it takes about 266 days. That means up to birth. To birth, it takes about 266 days. This is intrauterine life. In general, it is 266 days or 38 weeks if counting starts from fertilization or conception. If counting starts from the last normal menstrual period, the last normal menstrual period is called menstrual dating, and we add 14 days to the date of conception. 260 plus 14, totally 280 days, which is 14 weeks. So during our clinical practice, clinicians commonly use the menstrual dating rather than the conception dating, because the time of fertilization, the date of fertilization, it is difficult to know the date of fertilization. Because of that, we can estimate the gestational age using the last normal menstrual period, which is about 40 weeks. So in general, we assume 28 days. The normal menstrual cycle takes about 28 days, and ovulation on day 40. This is a normal. In general, the normal menstrual cycle can range from 21 to 35 days, not exactly. Some women's menstrual, the total menstrual cycle can vary from 21 to 35 days. If menstruation takes place, the menstrual cycle, if it takes 21 days, ovulation occurs in day 7. If menstrual cycle length is 28 days, ovulation day will be 14. And if menstrual cycles takes 35 days, ovulation day it occurs at 21 on day 21. So it varies. Ovulation date varies depending on the menstrual cycle length. So when before we discuss about the process of fertilization and gametogenesis, we will have some overview on the reproductive organs of the male and female. You will learn the detail of this in the respective modules, but I will give you here the overview of the male and the female reproductive organs. So the female reproductive organs, in general, is divided into two parts. The external genitalia, which includes the labia majora, the clitoris, and the labia minora, and the hymen, and the internal genitalia of the female, which includes the vaginal canal, the uterus, and the fallopian tube. And the uterus is further divided into three main regions. One is the cervix, the part which protrudes towards the vagina is called the cervix. The intravaginal part of the cervix. There is also extravaginal part of the cervix. And this part of the uterus is called the body of the uterus. And this, the most anterior part of the uterus, or superior part of the uterus, is called, above the entrance of the fallopian tube, is called the fundus. Fundus, body, and cervix, and the vaginal canal. And histologically, as you can see, the wall of the uterus. The wall of the uterus is divided into three layers. The first layer is the epithelial lining, or the epithelium and the lamina propria is called the endometrium, where implantation takes place. And this middle layer is called the myometrium. It is a muscular layer. It is made up of smooth muscle cells. And the outermost layer is called the perimetrium. The perimetrium. So in general, sperm, it swims or travels through the vagina, through the uterine lumen, and enters into the fallopian tube, and fertilization takes place here in the dilated part of the fallopian tube, which is called the ampulla of the fallopian tube. So this is the area where fertilization takes place. This is the anterior view, the posterior view, sorry, the posterior view. The previous one is the lateral view. The lateral view. This one is the lateral view. This one is the posterior view of the female reproductive structures, the vagina, the cervix, the wall, the body of the uterus, and the fundus of the uterus. This is the fundus. Above the entrance of the fallopian tube is called the fundus. This is a fallopian tube. It is further divided into different regions. The isthmus, the intramural part here, inside the wall of the uterus, there is intramural part of the fallopian tube. This is the isthmus of the fallopian tube. This dilated part of the fallopian tube is called the ampulla of the fallopian tube, where fertilization takes place. And this is the infundibulum. This funnel-shaped part is called the fundibulum, and these finger-like projections are known as the fimbriae, which captures the egg, which is released from the ovary. This is the ovary. Okay, the ovary. So this is, so as we have said, the wall of the uterus. The wall of the uterus has three layers: the endometrium, where implantation takes place, the fertilized zygote or embryo, it implants, it buries, buried within the endometrium, and it continues growth within the endometrium, and this is the myometrium, and this is the perimetrium. This is a smooth muscle layer. When you see the detailed structure of the endometrium, this pale area is the endometrium. When you see the detailed structure of the endometrium is histologically, it is further divided into first into 3 layers, the wall of the uterus into 3 layers, and the endometrium is further divided into functional layer and basal layer. This is the functional layer. This is the light microscopic appearance of the endometrium, having 2 layers, 2 distinct layers. The first one is the functional layer and the basal layer. The functional layer is a layer which undergo cyclic change in response to hormones, estrogen and progesterone. And if fertilization do not takes place, this functional layer of the endometrium will sloughed off, will be removed, and it's discharged through the vagina as menses or menstruation. So this is the functional layer. This pale area is called the functional layer, and this dark part here is called the basal layer, and this is the myometrium. The myometrium, within the functional layer of the uterus, the endometrium, there are spiral arteries and veins, and also endometrial glands that provide nutrients to the fetus during its early development. And the fallopian tube, as we have said, it has 3 parts, and fertilization takes place here at the site of the ampulla. This dilated part of the fallopian tube is called the ampulla, where fertilization takes place. Here the sperm and the egg unite here, and the fertilized egg or the zygote then starts to move towards the uterus, and finally it will be attached to the endometrium, and it will gradually penetrate towards the endometrium, and it will be buried inside the endometrium, and growth will take place. So this process is called implantation. Implantation normally takes place in the body of the uterus, in the posterior aspect of the body of the uterus, near to the fundus. Okay, that is a normal site of implantation. Sometimes implantation may take place in some other places, like towards the cervix, or implantation sometimes takes place in the fallopian tube. Okay, these are abnormal sites of implantation, and if it takes place in the fallopian tube, it is called ectopic pregnancy. And if the implantation takes place here in the cervix, and it's called if the placenta touches near to the cervix, it is called placenta previa. Okay, different types of placenta previa. So, but the normal site of implantation is in the body of the uterus, near to the fundus. This is the ovary. The ovary is another reproductive organ of the female, and it contains the egg. The egg cells are found within the ovary, and it is histologically, when you see the cross-section of the ovary, it appears like this one, and it has a central region that is called the medulla, which contains the blood vessels and lymphatics, and the peripheral dark area is called the cortex. The cortex, which contains oocytes or egg cells at different stages of maturation. And for example, here this is the primordial follicle, the primary follicle, the secondary follicle, the antral follicles, the Graafian follicle. The Graafian follicle is a follicle which is ready to rupture and release the egg. The process called ovulation. This process called ovulation. And as you can see, the egg is located at the center of the follicle, and the egg is surrounded by cells known as follicular cells. So the egg cell is not a naked cell. It is covered or surrounded by different layers of the follicles. Okay, if it is only one cell layer, either it's called primordial or primary follicle. If the layer increases, if it has many layers, it is called secondary follicle. And if it has some cavities inside the follicular cells, that follicle is called antral follicle. Antral follicle, at the center is the X. So this follicular development is stimulated by hormones known as follicular stimulator, FSH, and LH hormone, especially the LH hormone. It's the triggers ovulation, and FSH and LH hormones are produced by the anterior pituitary gland. So these FSH and LH hormones, in turn, regulated by another hormone produced by the hypothalamus, known as GnRH, gonadotropin releasing hormone. So any disturbance to this hormonal production results in abnormal growth or failure of growth of the follicles. Follicles may not grow from one stage to another stage in the absence of these hormones, these FSH and LH hormones, and finally ovulation may not take place, and pregnancy may not occur. And this is how some contraceptive hormone pills are working. After ovulation, the egg is released out to the outside, and it will be captured by the fimbriae of the fallopian tube and enter into the ampulla of the fallopian tube, where fertilization takes place. The remaining part of the follicle, it will remain inside the ovary, where it becomes corpus luteum, which is an endocrine tissue, which later produce progesterone if fertilization takes place. If there is no fertilization, this corpus luteum will regress, it will atrophy, undergo atrophy or degenerates and become corpus albicans. So this is ovary and its follicles. When you come to the male reproductive structure, mainly we focus on the testis. The testis is found within the scrotum. This is the penis. This is the scrotum, and inside the scrotum there is testis. Testis is the site where sperm is produced. The specific structures within the testis that produce sperm are known as the seminiferous tubules. This is a cross-section of the testis, the inside of the testis, which contains, as you can see, coiled tubes. These coiled tubules are known as seminiferous tubules. Seminiferous tubules produce sperm. Sperm-producing sites within the testis are called seminiferous tubules. Okay, this is all about the overview of the male and the female reproductive organs. Here, but before I go to the next portion, once the sperm is produced within the testis, it travels through a series of ducts. The first one is the epididymis, but before that there are efferent ductules, rete testis. This part is called rete testis. This part which receives the sperm directly from the seminiferous tubule is called rete testis. This is afferent tubules, and this is the rete testis, the efferent tubules, the epididymis, the coiled tubes known as the epididymis, and which are head, body, and tail, and then this is the ductus deferens. Then through the ductus deferens, sperm goes through the ductus deferens, and it goes through the and then to the ejaculatory duct, through the finally through the urethra. The urethra, the prostatic urethra, the membranous urethra, and the penile urethra. The penile urethra or the spongy urethra. This is different portions of the urethra. Then sperm is, once it is released, it enters into the vagina and it swims through the vaginal canal and into towards the cervix to the uterus, the uterine canal, and then the sperm goes through the fallopian tube. Finally, it reaches the ampulla of the fallopian tube where fertilization takes place. Once fertilization takes place, zygote is formed. The zygote is then start to move or travel toward this uterus to be implanted. But as it moves through the fallopian tube, it undergoes successive mitotic division, and the single-celled zygote becomes 2, then from 2 to 4, 4 to 60, and so on. And finally, this stage is called the morula stage, which is surrounded by a very thick, dense glycoprotein known as zona pellucida. This is zona pellucida. This thick glycoprotein covering is known as zona pellucida. And this embryo with cavity is called the blastocyst, named as blastocyst. Finally, the blastocyst will be attached to the wall of the endometrium, and it penetrates through the wall of the endometrium, and finally it will be buried, embedded within the endometrium. In general, developmental period is divided into two. In general, developmental period divided into prenatal, before birth, and postnatal, after birth. So there is also growth of the fetus after birth, that is postnatal growth, and before birth it is called prenatal. The prenatal period is further divided into two main periods: the embryonic period and the fetal period. So the embryonic period, it is a period during the 3rd to the 8th week of development is called the embryonic development, and from 9th to birth is called the fetal period. So most of the visible changes in the embryo takes place during the embryonic period. Whereas in the fetal period, it is differentiation and maturation process. The differentiation and maturation process. But new organs, new tissues are formed during the embryonic period. So that's why the embryonic period is a very vulnerable time at which the embryo can be affected by viruses and by different environmental factors. So the mother has to take appropriate precautions during the embryonic period. So different birth defects occur during the embryonic period. So the embryonic period and the fetal period. Okay, this shows the different, the two main periods of the perinatal period. The embryonic period, the first 8 weeks of post-fertilization. Major events, organs form from 3 primary germ layers. The 3 primary germ layers are the ectoderm, endoderm, and mesoderm. All the organs and tissues of our body develop from the 3 germ layers, known as ectoderm, endoderm, and mesoderm. These 3 germ layers are developed during the embryonic period, and organ systems also develop during the embryonic period. So it starts from fertilization, first week development, 3rd week embryo, it looks like this one, 5th week embryo, it looks like this one, and 8th week embryo, it looks like This one, at 12th week, the fetus, it appears like this, and that is the fetal period, which week 9 up to 38. Major events are organs grow in size and complexity. So in just 38 weeks from conception to birth, a single fertilized egg cell develops into a fully formed cell. What's the significance of embryology? As a physician, why you study embryology? One, embryology is allow you to for better understanding of the adult human anatomy. Moreover, embryology it helps to explain the origin of many birth defects. So you can explain if you have the knowledge of the normal growth of the intrauterine growth development, then you can easily explain the different types of birth defects and anatomical abnormalities. Embryology for better understanding of during your pediatric attachment, you know, obstetric attachment, you will appreciate the importance of embryology, starting from the time of ovulation up to birth. You will see different types of birth defects, and using your knowledge of embryology, you can explain how these birth defects occur. Okay, so how, as we have said, fertilization is a union of sperm and egg. How this egg and sperm are produced or developed. This process is known as gametogenesis. Gametogenesis, if it is for the egg cell, it is called oogenesis. If it is for the sperm cell, it is called spermatogenesis. So oogenesis is a sequence of events by which oogonia is a primordium of sperm egg cell, are transformed into mature oocyte. This maturation process begins before birth. Before birth, in females, it's before birth, and is completed after puberty. This maturation process, it goes up to menopause. Oogenesis, it occurs up to menopause, which is a permanent cessation of menses. Menopause means permanent cessation of menses. Menses means bleeding associated with menstrual cycle. So how this oogonium, from where this oogonium is derived? Oogonium is derived from the primordial germ cell. Primordial germ cells are germ cells found during the intrauterine life. This shows a 5-week embryo, where the primordial germ cells, which start from this site, start from the umbilical vesicles, from the 𝑦𝒏𝒕𝒓𝒐. This area is called the 𝑦𝒏𝒕𝒓𝒐. You will learn about the detail of this. Umbilical vesicles or the 𝑦𝒏𝒕𝒓𝒐, these are the origin of the primordial germ cells. During intrauterine life, these primordial germ cells, then they migrate through the umbilical vesicles and through the mesentery, and finally they will infiltrate or will be incorporated into the gonad, primordium of the gonad, the gonadal ridge. This is the gonadal ridge where the primordium of the germ cells migrate and infiltrate. These primordial germ cells later they develop in oogonia. Then the oogonia under successive mitotic process and meiotic process, it gives rise to the egg cell. Finally, the egg cell. So the egg formation starts during the intrauterine life of the embryo. Okay, oogogenesis starts before birth, and it will stay up to the stage of menopause. So this shows how this primordial germ cells. This is gonadal ridge. This is a ridge. This part of the embryo later develop into the testis and the ovaries. The ovaries, but the primordial germ cells, they migrate from the yolk sac, and incorporated into the ridge, into this gonadal ridge. So if this gonadal ridge, it develops into testis if the zygote carries the Y chromosome, and this gonadal ridge develop into the ovary if the zygote carries XX chromosome. So it is the Y chromosome that is responsible for the differentiation of the sex. Sex differentiation is due to the Y chromosome. The Y chromosome, it carries a gene. Okay, I will tell you this gene later on. So as the cortical cortex increase in size, the primordial germ cells are incorporated. They will be incorporated. At approximately 16 weeks of intrauterine life, these cortex began to break up into isolated cell clusters known as primordial follicles. So these follicles, these projections, known as the primordial germ, the mesenchyme, here the mesenchyme, and these are the finger-like projection. Later they break and form epithelial-like cells around the primordial germ cell. That primordial germ cell will be surrounded by epithelial-like cells and become the primordial follicle. And it then undergo further mitotic division. So it undergo mitosis, active mitosis. These cells undergo active mitosis, and after undergoing this mitosis, the follicles named as primordial follicles. So primordial follicle is a follicle which contains oogonia, or primary oocyte, surrounded by a single layer of follicular cells. So this is how it happens. So this is the oogonia, the primordial germ cell, surrounded by cells which derive from these projections. That follicular cells are derived from these follicular cells, as you can see, they are derived from surface epithelium. So this portion of the ovary later it develops into cortex, and this portion of the ovary later it develops into the medulla. So in general, oocyte maturation, it has two stages: prenatal maturation, that is maturation before birth, and postnatal maturation, maturation after birth. So primary oocytes and primordial follicles. So as we have said, that during early fetal life, oogonia proliferate by mitosis. Then the cells will enlarge to form primary oocytes. This takes place before birth. So primary oocytes. These primary oocytes derived from the primordial germ cells. As the primary oocytes forms, connective tissue cells surround it and form a single layer of latent follicular epithelial cells. The primary oocytes enclosed by this layer of cells constitute primordial follicle. So this is the event that takes place before birth. First, there is oogonem. Thus, this oogonem or stem cell, which is derived from primordial germ cell, undergoes successive mitotic cell division and become primary oocyte. The primary oocyte is then it will be surrounded by single layer of follicular cells and the whole structure is known as primordial follicle. So this is a process that takes place during before birth. Then the primary oocytes undergo further growth postnatally after birth. After birth, the primary oocyte before birth, the primary oocyte begins the first meiotic division. Because gamete formation, it involves meiotic cell division. After mitosis, the primary oocytes then undergo meiotic cell division. But the meiotic cell division will not be completed. It will be arrested at prophase 1. Prophase 1. So meiosis starts before birth, but it will not be completed, and it will be arrested at prophase 1. So this completion of meiosis 1 takes place after puberty. After puberty, when the egg cell starts to ovulate. Near the time of ovulation, prophase 1, the cell will change from prophase 1 to metaphase 1, then it continues. Okay. So primary oocytes, it undergo meiosis 1, but it will not be completed, and it will be arrested at prophase 1, and meiosis 1 will be completed after puberty, which is during the time of ovulation. So, so the ovary during infancy and childhood, the ovary is still contains primary oocytes and primordial follicles. So we don't have primary follicles and secondary follicles before the time of puberty. So during infancy and childhood, the female ovary contains only primordial follicles. Primordial follicles which are arrested at prophase 1. At prophase 1. So each month from puberty, from puberty to menopause, one follicle starts to mature each month, and ovulation occurs. As the primary oocyte enlarges during puberty, the follicular epithelial cells change their size and become cuboidal. So primary oocyte surrounded by cuboidal epithelial cells is known as primary follicle. Previously, it is called primordial follicle. Now, when the follicular cells change into cuboidal, then it is called primary follicle. The primary oocyte soon becomes surrounded by a covering amorphous acellular glycoprotein material that is called zona pellucida. Zona pellucida. As the follicles mature, the primary oocytes increase in size, and shortly before ovulation, it completes the first meiotic division to give rise to secondary oocyte and the first polar body. So during the time of ovulation, near the time of ovulation, the primary oocytes will undergo further meiotic division from prophase 1 to metaphase 1, and divide and completes meiotic division. So meiosis 1, during as a result of meiosis 1 will be completed after ovulation. The secondary meiosis 1 results in the formation of secondary oocyte plus first polar body. Then the secondary oocyte receives almost all the cytoplasm, and the first polar body receives very little. So in general, the first meiotic division results in the formation of two cells. One is the secondary oocyte, the other one is the vestigial cell known as the first polar body, which later degenerates. But the secondary oocyte continues the second meiotic division, but that second meiotic division will not be completed. Again, that second meiotic division will be arrested at metaphase 2. So meiosis 2 will be arrested at metaphase 2, and meiosis 2 will be completed after fertilization. When the egg is fertilized with the sperm, then meiosis 2 will be completed. That means if the sperm penetrates the secondary oocyte, the second meiotic division is completed, and the most cytoplasm is again retained by one cell, the fertilized oocyte, the other cell, the second polar body. So fertilized oocyte plus secondary polar body will result as a result of meiosis 2. So meiosis 2, if there is no fertilization, meiosis 2 will not be completed and it will be arrested at metaphase 2. So it is fertilization that initiates further the cell to undergo further meiotic 2 division. Then the polar body will be extruded, maturation of the oocyte will be completed. As you can see, this summarizes the stages that occur from puberty to menopause. From puberty to menopause, we start with the primary oocyte, which is arrested at prophase 1. And this prophase 1, that's meiosis 1, will be completed as a result of fertilization. So the primary oocyte, when it is surrounded by a cuboidal epithelium, single layered cuboidal epithelium is called primary follicle. If it has multiple layers of cuboidal follicular cells, is known as secondary follicle. The secondary follicle, again, it is surrounded by a glycoprotein covering known as zona pellucida. So once fertilization takes place, meiosis 1 will be completed after ovulation, near the time of ovulation. Then the secondary oocyte here, this is the primary oocyte. It divides into two. First secondary oocytes and the first polar body. And the secondary oocyte, until fertilization, it will be arrested in metaphase 2. And when fertilization takes place, this is a sperm. When fertilization takes place, the secondary oocyte will continue meiotic 2 division, and it will metaphase 2, then anaphase, telophase, and then it divides into another 2 cells. The second polar body and the fertilized oocyte, ovum, which is fertilized by the sperm. So once fertilization takes place, the oocyte will complete its maturation and it completes its meiotic 2 cell division. Okay, so this is this is a follicular change in the ovary. This is changes involving the oocytes. So once the oocyte is matured, then the ovary, the follicles will rupture and release the oocyte. The oocyte will be released and it will be fertilized. And if fertilization, in the presence of fertilization, the follicle, the remaining part, the remnant of the follicular cell, it will become corpus luteum, which will later produce progesterone. So in general, the ovaries contain approximately 2 million primary oocytes or primordial follicles in the ovaries. That is, a newborn female contains about 2 million primary oocytes. So no further production of primary oocytes after birth. So a female born with a limited number of primary follicles or primordial follicles. These primordial follicles, starting from the time of fertilization, or ovulation or puberty, they will start to deplete. Depletion of the primordial follicles takes place. The follicles in general takes place until menopause. And by the time the female reaches to the stage of menopause, the entire follicles will be depleted and the female will not have other follicles to ovulate. So, but most as development progresses, most of the follicles will disappear. During childhood, and adolescence, only 40,000 follicles will remain. From out of these 2 million primary oocytes, by the time of childhood and adolescence, only 40,000 follicles will remain. So there is follicular degeneration from birth up to puberty. Throughout the female's life span, there is follicular degeneration, and by the time of adolescence, only 40,000 follicles will remain. Out of these 40,000 follicles, only approximately 400 become secondary oocytes and are expelled at ovulation during the reproductive period. The rest will degenerate. The rest of the follicles will degenerate. So only 400 follicles will ovulate until the life of the during the reproductive period of the woman. Few of these oocytes, if any, are fertilized and become mature. Okay. The number of oocytes that ovulate is greatly reduced in women who take oral contraceptives because the hormones in them prevent ovulation from occurring. So oral contraceptives, these are hormonal contraceptives that disrupt the hypothalamus, hypophyseal ovarian axis, hypophyseal pituitary ovarian axis disruption. It will be disrupted by these exogenous hormones, the oral contraceptives. As a result of that, ovulation may not be takes place, and that will result in absence of pregnancy. So as I have shown you, these are the follicles in the cortex of the ovary, primordial follicles, primary follicles, secondary follicles, antral follicles, and graphial follicles, and finally the corpus luteum and secondary oocyte. And these follicles, as they grow from one stage to another stage, most of the follicles will degenerate, and only few of the follicles will remain throughout the reproductive life of the woman. So this is once the egg is ovulated, it will be surrounded by remnants of some follicular cells known as corona radiata. Corona radiata. These remnants of the follicular cells. Okay, the ovulated egg, it looks like this one. Okay, it looks like this one. There is at the center there is nucleus. This is the cytoplasm of the oocyte, the cell membrane, and this is another layer called zona pellucida, a glycoprotein layer, and these are remnants of follicular cells known as the corona radiata. So this is the structure of the ovulated egg. So ovulation, when the egg ovulates, it's not naked. It is surrounded by different types of structures like zona pellucida and corona radiata. Then the sperm penetrates these layers and injects its genetic material into the nucleus of the oocytes. So that is all about the development of the egg cell, oogenesis. In the case of males, there is it's called spermatogenesis. Spermatogenesis, unlike oogenesis, spermatogenesis, it takes place throughout the life of the individual. As we have said, in females, there is limited number of egg cells in the ovary, and these egg cells will be gradually depleted, and by the age of menopause, the female will not have any egg cells or any follicle to ovulate. But this is different in the case of males. In males, there is continuous production of primary spermatocytes. There is continuous production of primary spermatocytes throughout the life of the individual. But in the case of females, there are limited number of primary oocytes. So where this spermatogenesis takes place? It takes place in the testes. The testes of the individual. This is also another difference from that of the female. In female, oogenesis takes place until the time of menopause. After menopause, there are no follicles in the ovary, so there is no ovulation and there is no oogenesis. But in the case of males, spermatogenesis takes place throughout the life of the individual. After several mitotic divisions, the spermatogonium grow and undergo changes. So sperm starts to grow from a primordial cell or precursor cell known as spermatogonium. For females, it is called oogonium, spermatogonium. Where do you find this spermatogonia? These spermatogonia are found within the wall of the seminiferous tubules. Within the wall of the seminiferous tubules. Because this is the testis where sperm is produced. And these are the specific sites, areas inside the testis, these convoluted tubules, coiled tubules within the testis are called seminiferous tubules. These are sites where sperm is produced. So the wall of the seminiferous tubules, when you examine the wall of the seminiferous tubules, we can identify two types of cells. These orange colored cells are known as Sertoli cells, the supporting cells. These cells are the oogonia. Okay, this is the basement membrane. This is the wall of the seminiferous tubule. This is the wall of the seminiferous tubule. In the wall of the seminiferous tubule, there are two types of cells. One is this orange colored cells known as Sertoli cells, and these are the spermatogenic cells, which later develop into the sperm cell. So initially we have the precursor cells or primordial cells adjacent to the basement membrane known as spermatogonium. There are two types of spermatogonium, type A and type B. Type A and type B. The type A will remain there and it will maintain the pool. Okay? It will remain there and give rise to the secondary, the other type of oogonia known as type B. Spermatogonium. This type B spermatogonium is the one which undergo meiotic division and finally give rise to the sperm cell. Whereas type A will remain there and it serves as primordial cell. So there is oogonium, then there is primary oocyte, secondary oocyte, spermatids, the late spermatids, these are early spermatids. These are the late spermatids, and finally, these late spermatids will detach from the Sertoli cells and become matured sperm cell. So this is a continuous process, and these daughter cells, unlike the oogonium, this spermatogonium, unlike the oogonium, they are continuously produced throughout the life of the individual. But in the case of oogonium, it is the oogonium in females is limited in number. So these are the stages of spermatogenesis: spermatogonium, primary spermatocyte, secondary spermatocyte, early spermatids, and late spermatids. Okay, the process of spermatogenesis is divided into 3 phases: spermatocytogenesis, meiosis, and spermiogenesis. Spermatocytogenesis is differentiation of spermatogonia into primary spermatocyte. That's the first stage. The spermatogonium differentiated into primary spermatocyte. This is through successive mitotic cell division. The second stage is meiosis. Meiosis, reduction division, whereby the diploid primary spermatocytes reduce their chromosome number and forming haploid spermatids. And here we have meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. The cell which undergo meiosis 1 is called primary spermatocyte. Primary spermatocyte, it undergo meiosis 1 and results in 2 cells, 2 daughter cells, with haploid number of chromosomes and with sister chromatids. And meiosis 2 will result in division of the sister chromatids into 2 and result in another 2 cells. So totally we have 4 cells produced from 1 primary spermatocyte. The other one is the last stage is spermiogenesis. Spermiogenesis, it refers to transformation of spermatids into spermatozoa. Okay, so after completion of meiosis 2, the cells are called spermatids. After meiosis 2, the cells are called spermatids. These spermatids undergo a process known as spermiogenesis. Spermiogenesis means transformation of the spermatids into spermatozoa. So this is how the process takes place. This is the Sertoli cells. These are the Sertoli cells. By the way, these Sertoli cells are give protection, support, and nutrition to the spermatids, to the spermatogenic cells. The spermatogenic cells. So during the first stage, as we have said, this is the spermatogonium, which undergo mitotic division and give rise to primary spermatocyte, each having 46 chromosomes. And then these primary spermatocytes undergo first meiotic division, give rise to haploid number of cells with haploid number of 2 cells, haploid number of chromosomes, each with 23 chromosomes, and each of these cells further undergo second meiotic division, resulting in the formation of 4 cells with haploid number of chromosomes. Here, the second meiotic division, it involves separation of the sister chromatids. Here, it is separation of the homologous chromosomes. First meiotic division is mainly, it involves, it is a process of separating the homologous chromosomes, whereas the second meiotic division, it separates the sister chromatids. So in general, we have 4 cells at the end of meiosis, meiosis 1 and 2. And these cells, in general, they are called spermatids. As you can see, the spermatids are oval-shaped cells with no tail, neck, and head. But these spermatids further undergo structural modification and to become mature sperm cells. Okay, this is what I have already explained. See, this summarizes the whole process. So we have spermatogonium, 2 types of spermatogonium, type A and type B. The type A is the one which remains there as a precursor cell, as a primordial cell, whereas the type B is the one which continues growth by meiosis, first by mitosis, then through meiosis. So this is called the wall of the seminiferous tubule. As you can see, it is divided into 2 parts, the basal compartment. This is a gap junction. After the gap junction, the 2 Sertoli cells are here interconnected by a gap junction. So after the gap junction, the area between the gap junction and the basement membrane is called basal compartment. This basal compartment, it contains only the spermatogonium. The spermatogonium never cross towards this compartment. This compartment is known as the luminal compartment. This is the luminal compartment. So what are the process of spermiogenesis? So spermatids differentiate into sperm. The process of differentiation of spermatids into mature sperm cell is called spermiogenesis. So spermatogenesis and spermiogenesis are two different processes. Spermatids discard much of their cytoplasm, rearrange their organelles, and form a flagella or a flagellum to become transformed into spermatozoa. So in order to transform to spermatozoa, the spermatids should discard much of their cytoplasm. So the sperm cell needs only small portion of the cytoplasm of the cell. Most of the cell is occupied by the nucleus and the other organelles. And the cell also form tail. Spermatids form the tail. Okay, so this structural rearrangement or modifications of the spermatids is known as spermiogenesis. So in general, the spermiogenesis is subdivided into 4 phases. The first one is the Golgi phase, the cup phase, and the acrosomal phase. Okay? The Golgi phase, the cup phase, and the acrosomal phase. Okay, the Golgi phase. Break እንውሰድ። ስንት ሰዓት ላይ ነው ብሬክ ተወስዱት? 4 ሰዓት ሆኗል። እስኪ ምልክት አሳዩኝ if you need break. Okay, so after having some 30 minutes break, we'll be back and we'll finish the remaining part.